We've been talking about the levels of organization within a muscle. We have the entire muscle, which is made up of fascicles. Fascicles are made up of muscle fibers. Muscle fibers are made up of myofibrils. So what are we looking at in this picture right here? In this illustration, what is the entire thing in that picture? You can go ahead and cheat and look at the caption at the bottom. That's one muscle fiber. That entire illustration there is one muscle fiber, which of course is the same as one muscle cell. You are looking at a single muscle cell or a single muscle fiber. So what are all those big things that it's made up? Those big things that look like, hot dogs sort of? Those are the myofibrils, all right? See how important it is to make sure you know what you're looking at here? So there are thousands of transverse tubules that permeate the sarcolemma. Remember what the sarcolemma was? The sarcolemma is the cell membrane of a muscle cell, which is the same as a muscle fiber. So when you look at the far left of that illustration, you see that blue stuff. It kind of looks like a shiny blue stuff. That's the sarcolemma. Now the endomysium would be outside of that. So the endomysium is the layer of the deep fascia. That's connective tissue. Now we're looking here at that light blue on the far left. That's the sarcolemma. That's epithelial tissue. That's a membrane, all right, that covers a single muscle fiber or muscle cell. Now there are openings that lead, that are the openings to tubes, and the tubes lead down inside. Those tubules are called transverse tubules, and we can usually just abbreviate them as T-tubules. So what's the big deal with that? Why do we have that? Well, remember, muscles don't contract by themselves. What causes a muscle to contract? Electricity, all right? So what happens here is that a muscle action potential, an MAP, will travel along the sarcolemma and go down the T-tubules. So as we'll see coming up eventually, these muscle action potentials are ultimately created by nerves, all right? So nerves, remember, electrical tissue. So what's going to happen is the nerves generate the electrical impulse. That electrical impulse then travels down the sarcolemma. It flows along the sarcolemma, electricity flowing along the outside of the muscle. And then it goes down these T-tubules, and that's how the electricity gets inside of the muscle in order to make it contract. Inside of the muscle fiber, let me be specific, because we're looking at a single muscle fiber here. We need the electricity to get inside to make it contract. So all those big hot dog looking things there in the picture, those are the myofibrils. Now what exactly is a myofibril? It's the contractile organelle of a muscle cell. Stop and think about that for a moment, because I'm going to ask about this on quizzes and exams, and it always kind of amazes me how many people don't know this or seem to have forgotten it. So remember cells. Cells have all kinds of organelles, don't they? They have a cell nucleus, they have mitochondria, they have the rough endoplasmic reticulum, the smooth endoplasmic reticulum, the Golgi apparatus, the lysosomes, the peroxisomes. Well, muscles have their own unique kind of organelle. No other kind of cell has this organelle. And what organelle am I talking about? A myofibril. And what is it that myofibrils do? They can contract. Remember what makes muscle tissue different than any other tissue in the body? The fact that it can contract. Well, how does muscle tissue contract? It's ultimately the myofibrils, the contractile organelles. Those are what make the muscles actually contract, all right? And that's how you move and do things. Myofibrils make up roughly 80% of the total cell volume. So that's mostly what a muscle cell is. It's mostly just a whole big bunch of myofibrils, because that's the main job muscles have, is to move, to contract. And so myofibrils are what does that. Now, the myofibrils are encircled by something called the sarcoplasmic reticulum. So we've seen before endoplasmic reticulum, both smooth and rough. Reticulum, the Greek root word ret, means a net. So anytime you see that root word, it means that something is net-like. So the sarcoplasmic, why is it called that? Well, because it's inside of the sarcoplasm. Remember, the sarcoplasm is the goo inside of the muscle cell. So what we do is we have a big net that's in the sarcoplasm. It wraps around the myofibrils. So in the illustration on the left, see those kind of those Dijon mustard looking lines? I'm trying to use my Project Runway terminology. So see those lines that are running on the outside of the myofibril? That's the sarcoplasmic reticulum. Now, what's the big deal about it? Why is it important? Because that's where we store calcium. And muscles cannot contract without calcium. Remember when we did bone homeostasis, we talked about hypercalcemia and hypocalcemia? Remember what the signs and symptoms were? Hypercalcemia, you had muscle weakness. Hypocalcemia, you had tetany, convulsions, things like that. Muscles were contracting, spasming. So yeah, calcium has a lot to do with muscle contraction. So muscles need tons of calcium, and so they keep it right on hand. Remember how I said that muscles had, what were the two things that muscles had for oxygen and glucose, that they kept it right there inside? Right, glycogen and myoglobin. Yeah, well, in addition to needing tons of oxygen and tons of glucose, muscles need tons of calcium. So they store the calcium in the sarcoplasmic reticulum. That's what it's for. It's to store calcium. And what happens is, if you kind of look at the diagram there, so you can see those blue T tubules coming down from the sarcolemma. Then notice also, closer to the right side of the illustration, you can see the T tubules going around the myofibrils. So right next to the blue T tubules, you can see there's the Dijon mustard colored sarcoplasmic reticulum. Notice as the sarcoplasmic reticulum gets right up next to the T tubule, it also forms tubes that run parallel to the T tubule. Those are called terminal cisterns. Terminal because they're at the ends of those bands of sarcoplasmic reticulum. Terminal means at the end. And cistern, as you may know, a cistern's like a big tank for like holding water. Here in Tucson, back before, back in the 1800s, there was no, city water supply. You had a big tank and you let it fill with rainwater all year long, and that's what you lived on for your water. So terminal cisterns, again, are the Dijon mustard colored tubes that are running right next to the blue T tubule, and they are full of calcium. So that's the entire sarcoplasmic reticulum is full of calcium, but the calcium is going to actually get released from the terminal cistern. That's the big deal about the terminal cisterns. It's where the calcium is released. And then myofibrils are in turn, that's not even the smallest unit yet, they're made up of thick and thin filaments. And you can see, if you look carefully at that illustration, you can see towards the end, see how thick and thin filaments are labeled? Yeah, so they're a bunch of overlapping filaments. Some of them are thick, some of them are thin, and that's what's actually going to cause the contraction to take place inside the myofibril. So the thick and thin filaments don't run the entire length of the muscle. See how they stop? And then see how there's a zigzag line on the end? See there's zigzag lines on both ends? Those zigzag lines are called Z-discs because it looks like a whole bunch of Z's all connected together. So they mark, basically, there are little compartments within the myofibril. Little compartments made of thick and thin filaments. Each one of those little compartments is called a sarcomere. So you can see that labeled in the illustration. The sarcomere is one unit of thick and thin filaments. Each end is bordered by a Z-disc, okay? So just look at that for a little bit. Make sure you know the parts, okay? Here you see a nice illustration showing the whole deal here. The whole enchilada. So in the upper right, you got a muscle fiber. So all those little things inside would be myofibrils, right? And then on the bottom, you see it little zoomed in. We're now going to zoom in on a single muscle fiber. We're going to see how it's covered by sarcolemma, right? And it's got all that sarcoplasmic reticulum. Here now, the sarcoplasmic reticulum is all the blue stuff. And then the T tubules now are the yellow ones, the regular mustard kind of yellow. All right, and then when you look carefully at a single myofibril there on the left, you can see it's made up of thick and thin filaments. The thick filaments are the blue ones, and the thin ones are the little red ones, okay? So were you paying attention? So, look in the upper left, the organelle. What organelle are we looking at? What is that? Which organelle of the muscle is that? Those are mitochondria. Remember, muscle fibers got tons of mitochondria because they're making tons of ATP. All right, below that, the membrane. What membrane are we looking at there? We're looking at a membrane that goes around the entire muscle fiber. So what is that? That would be the sarcolemma, okay? Then on the far left, what organelle is that? That whole thing sticking out, what organelle is that? That's a myofibril. That is the contractile organelle. And then moving along, what's that red structure? That's a thin filament. The blue one is a thick filament. Notice then see where it says triad? Triad, see in the middle is the yellow T-tubule, and then see the blue tubes on either side? Those would be the terminal cisterns, okay? Let's continue then. The structure, it's pointing at that blue network of stuff. That would be the sarcoplasmic reticulum. Then next, the structure, it's pointing at that yellow tube. So what's that? That would be a T-tubule. Then continuing to the right, the organelle. What organelles are those? Those are myofibrils. What fluid fills the muscle cell? Yes, sarcoplasmic. And once again, what membrane is that? That's the sarcolemma. And what structure do we see there at the end of the sarcoplasmic reticulum, running right alongside that yellow T-tubule? That is a terminal cistern. And they put it into the Latin terminal cisterna. You can just say terminal cistern, and I will be happy, okay? So take a look here, once again, looking at all the different parts. I keep emphasizing this, don't get mixed up. Upper left, that's a skeletal muscle with a fascicle sticking out. Then in the middle, there's a fascicle with a muscle fiber sticking out. Then on the bottom, there's a muscle fiber with a myofibril sticking out. And in the upper right, there's a myofibril, and you can see it's made up of lots of thick and thin filaments. Boxes inside of boxes, inside of boxes, inside of boxes. Keep them straight, kids. Okay, let's take a look here. There are 2 thin filaments for every thick filament. All right, see in the bottom part there, you got the big thick filament, and then there are the smaller thin filaments on both sides of it, one above and one below. Filaments overlap, creating the appearance of the zones and bands, which are the striations. Remember we said the skeletal muscle is striated? It had those stripes. What caused the stripes? It's basically the overlap of the thick and thin filaments. They create the bands, which, you know, up close they look like big wide bands. From a distance, they look just like little stripes. Those were the striations of skeletal muscle. So the dark middle part is called the A band, all right? So you can see there in the bottom, the A band. That runs the length of the entire thick filament. But notice it also contains some of the overlapping thin filaments. The thin filaments overlap the thick just a little bit when the muscle is at rest. So the A band is the length of the thick filament, but notice there are little bits of thin filaments in the A band. Then near the ends of the A band are the zones of overlap with both thick and thin filaments, which is what I just talked about. When you look at the striations of the muscle from far away, you see the dark stripe and then you see the lighter part. Just remember that the A band is the dark stripe, all right? And that's easy to remember because the word dark has the letter A in it, all right? So A is for dark. Now then, we can, next to the zones of overlap are the I bands, which are made up only of thin filaments, but not thick. So see there in the bottom, see where there's just the blue, all right? So where there's blue, but it's not overlapping the thick filament, that part is by itself is just called the thin filament, all right? And that's the I band. The I band, this is again easy to remember because the word light has the word I in it. So when you look, see in the upper left, see how it looks like that muscle is striped? It has the dark and the light bands. See, the dark bands are the A bands, the light bands are the I bands. The dark bands are made up of thick filaments plus some overlapping thin filaments. The light, the I band, is only thin filaments, okay? The H zone is the part in the middle, all right? It's right in the very smack dab in the middle, all right? So you can see in that middle illustration, the H zone, that contains thick but not thin filaments. So that's the part where there's only a thick filament but no overlap of thin filaments, called the H zone. And then in the middle of the H zone are some proteins that hold the whole thing together. The proteins are myomecin, and therefore we call the line in the middle, there's a line of these proteins. The proteins form a band or a line. We call that the M line. M is for myomecin, but you can think of it as being the middle because the M line is in the very middle of the sarcomere, okay? So look in the middle diagram. See the Z discs? Those mark the boundaries. Those are the ends of the sarcomere, all right? And see how there are places in where the Z disc is, where there's only light bands, there are only thin filaments? That would be the I band, all right? Then we come to the darker band. Notice it's made up of both thick filaments and overlapping thin filaments. So that's the A band. In the middle of the A band, where there's only thick filaments, that's the H zone. And running right down the middle of the H zone is the M line, which are those myomecin proteins. I know it's all new and weird, you just got to get used to the terminology. Z discs are stabilized by a protein called titin. So I don't think that's a huge thing, but it might show up, so you should probably know that. Z discs are made of the protein titin. M lines are made of myomecin. Z discs are they're stabilized by titin. So here you see a side view of muscle fibers, thick and thin filaments, not muscle fibers, sorry. But there's a muscle fiber in the upper left. But looking at myofibrils and now looking at all the thick and thin filaments, you may know that when you prepare slides, you have to stain them, otherwise you can't see anything. So what they did here is they've used different stains. You can see that in the upper right. So one of them stains the thin filaments, another one stains the thick, and then they stain both thick and thin. And you can see how basically they all form this giant network, the thin and the thick filaments all surrounding one another, and that's what's going to make the muscle contract. So just take a moment and look at these pictures. Start to get used to this in your head. So here you can see thick and thin filaments. That stuff that's, so what color is that? Is that teal? I'm going to go with teal for right now. I know I'm wrong. I know a whole bunch of you were sitting at home going, that's not teal, for God's sakes. That's, I don't know what, aquamarine or sea foam or I don't know what. Cut me some slack here, will you? But that would be the sarcoplasmic reticulum. You can see the T tubules, okay? Just on your own, spend a few moments, a little bit of time, just looking at these. Here, now that's definitely sea foam, okay? That would be the terminal cisterns. There you can see sarcoplasmic reticulum, you can see the T tubule, you can see the striations, the light and the dark bands, okay? So, and here, this picture really shows the whole thing. Holy smoke. It's got all the bands and everything, and the filaments. Oh my gosh, shows the SR. Holy smoke. Notice it's kind of like, SR is kind of like those things that some of you wear on the weekends. I've seen you down there at IBT and at Zen Rock and stuff. Oh, those places aren't even open anymore, are they? So yeah, so there you go. Good study slide. Look at all that stuff. And here, notice what's going to happen. So the thick and the thin filaments overlap each other, right? Well, when the muscle contracts, they slide past one another. That's what the contraction of muscle is. So look on the far left, you can see the cartoon version first. Go from top to bottom. You're looking at a muscle contracting. Look at how in the top, the thick and the thin filaments are only partly overlapping. Then in the middle, see how they're overlapping more? See how on the bottom they're completely overlapped? Yeah, exactly. That's how muscles contract. The thick and the thin filaments slide past one another. We call that the sliding filament mechanism. Now, back when I started college, they didn't even know that yet. They thought that the muscle, kind of like imagine you're taking a sheet of paper and you're folding up the paper. They thought that muscles contracted by the whole sheet of the muscle folding. They called that the accordion mechanism. We learned during my lifetime that that was wrong. It's the sliding filament mechanism. A lot of stuff that we know now, it wasn't that long ago that we learned it. I know you think we've always known everything, but we haven't. They keep finding new things all the time. And by the time, you're in your 40s and 50s and stuff, whole bunches of new things will be learned by then too. In the healthcare professions, you're never done learning. You have to keep learning your whole life because they keep updating. We keep upgrading as we learn more and more stuff. So take a look, read this through. During muscle contraction, the thin filaments are pulled over or past the thick filaments towards the M line, meeting and even overlapping in the middle of the sarcomere as the Z discs come closer together and the sarcomere shortens. So when a muscle contracts, the entire muscle gets shorter. And yeah, that's because as the thick and thin filaments overlap, each sarcomere gets shorter. So muscle is made up of thousands and thousands of sarcomeres. So as each one of those thousands of sarcomeres gets shorter, it causes the entire muscle to get shorter. So again, in that diagram on the left, look now at the actual photomicrograph. You can actually see those filaments from top to bottom overlapping one another. Look at the Z discs. See how far apart the Z discs are on the top? Now look on the bottom. The Z discs are closer together. The sarcomere just got shorter. And when all the sarcomeres get shorter, the entire muscle gets shorter, and that's how muscles contract. So it's the sliding filament mechanism. No accordions, okay? Give me all your cash. I have an accordion, and I know how to use it. Me too. I had a friend one time, he was an accordion player, and he went downtown just like on a weekend night, just clubbing, bar hopping, Club Congress, the Surly Winch, Chays, some of the stuff that used to be there, Plush. And then halfway through the evening, he remembered that he had left his accordion sitting in the back seat of his car. And he thought, oh my God. He went running back to his car. Sure enough, the back window was broken out, and there was another accordion inside of his car.